So it's a real pleasure to be here. Very excited to be here, and thank you very much for the invitation. Really pleased to be here with my good friend and colleague Emily, who's a wonderful partner on the patient side of things. And I'm going to tell you a little bit about Taysha Gene Therapies. We're a company that focuses on developing gene therapy treatments for rare and severe neurological diseases, in particular of children. I'm going to tell you a little bit about Taysha and our approach at Taysha, and then I'm also going to talk to you a little bit about the program we have specifically for Angelman syndrome. And we actually have two programs in development. You may have heard from our academic partners earlier in the meeting, and I'm going to touch on their programs. And then I'm happy to take questions if there's time for questions. Emily will also spend a few moments talking about partnerships in general and how we work with the patient community. So looking forward to going through the next 20 minutes or so. Next slide, please, Emily. We have a legal disclosure statement. I won't read through it, but it's, you know, our corporate lawyers say we have to have this with pretty much every presentation now. So essentially, it's to make the point that, you know, this is an educational presentation more than anything. Next slide, please. And I'll let Emily introduce herself when she comes on stage, and thanks Alison for the nice introduction. But yes, I'm a pediatric intensive care specialist by training, so I did actually work in clinical practice for a number of years. I trained in the UK and in Australia and looked after many children with rare, severe neurological and metabolic diseases. I then moved into industry. I'm boarded in pediatrics, internal medicine, pharmaceutical development, and I also have a degree in translational science. And most of my career in clinical research has been in pediatric, neurological, and metabolic disease. And more recently, I've spent a lot of time on the gene therapy side of things. Next slide, please. And one more. So the foundation of Taysha, and Taysha is a gene therapy company that started really close to 2 years ago, and it's been on a very rapid ascent. We actually launched the company really in earnest in the early parts of the pandemic, and our focus is really on leveraging proven gene therapy platforms. So you'll all be familiar with AAV9 gene therapy for spinal muscular atrophy, Zolgensma, and we really take a lot of that technology, the AAV9 capsid, the HEK293 mammalian cell suspension, and we use that across our range, across our portfolio of pediatric neurological diseases. We have a foundational relationship with the University of Texas Southwestern, and this is led by two individuals, and I'll speak a little bit more about the partnership in a moment, but led by Stephen Gray, who's a very eminent gene therapy researcher, and Burj Manassian, who is the division chair of pediatric neurology, also a very eminent clinical researcher. And we have a really wonderful collaborative partnership with UT Southwestern. And they essentially do a lot of the early proof of concept discovery work and then transition over to us as we move through into preclinical IND enabling studies in order to move the program into clinic. And then we also have a lot of gene therapy experience. I don't have the slide of our executive team, but most of us have had experience both in rare disease and in severe neurological diseases, and also in gene therapy, many of whom worked on SMA and Zolgensma, Emily being one of them, of course. Next slide, please. Now, there are three main tenets that we really focus on across our range of programs. And this is one of the values, one of the benefits of the approach at Taysha. So a lot of people ask us, you have lots and lots of programs. How can you do so much? How can you do so many programs? Well, a large part of the reason we can do that is because we have many elements that are common from program to program. So, for example, everything we do is AAV9 capsid. We don't do anything apart from AAV9 capsid. So AAV9 is a serotype that has the most clinical experience. There's over 1,500 patients on commercial gene therapy for SMA, and there's a lot of clinical trial precedent with AAV9. We know how to make it, we know how to scale it, we know that it gets into neurons and brain cells as it needs to, and so we focus specifically on that. On the manufacturing side, we use what's known as HEK, which stands for human embryonic kidney, 293 mammalian cell suspension process. Lots of words there, but basically it's a mammalian cell system. There are several different systems that people use to make gene therapy. So baculovirus is one, herpes simplex is another. But we feel most comfortable with the HEK 293 process, and that once again has the most precedent. If it works, we don't want to change it. And then the third foundational kind of element to what we do is we give all drugs intrathecally, which is with a small needle at the base of the spine. So around about L4, L5 at the back of the spine. We have the patient on their back. We inject the drug, turn the patient on their back, head down a little bit for about an hour, and we allow the cerebrospinal fluid to bathe the brain and the spinal cord with the gene therapy. And there's once again clinical precedent with that approach, not just in gene therapy. So with our giant axonal neuropathy program, CLN3, CLN6, which Amicus are working on, and zolgensma intrathecal, but also just in general. I actually gave a lot of intrathecal drug myself in my pediatric practice, not gene therapy, but oncology drug and an anesthetic drug. And we know that there's a good route of delivery to get to the brain and spinal cord. So we focus on those three things: AV9, HK293, and intrathecal delivery. That's common from program to program. Next slide, please. Just to touch on our partnership with UT Southwestern. So Stephen Gray's the gentleman in the lower left-hand corner, a very well-known, highly published, highly regarded gene therapy expert. Also a very nice person, very collaborative, very practical-minded. And he really, with Burge and ourselves at Taysha, our intent is to get these gene therapy treatments into patients as rapidly as possible and then commercialized as rapidly as possible, so to help individuals all around the world as much as possible. So, if things are good enough to get into clinic, as approved by the regulators, we'll move forward fast into clinic. Burge is his division chief of pediatric neurology, very eminent researcher, has done a lot of work in a lot of really very valuable trailblazing work in Rett syndrome in particular. Also, they've built out the largest pediatric neurology training group in the country. So a whole team of people below Burge and Steve who are very willing and able to help us and partner with us. And as I say, the proof of concept work, the proof of principle work in the animals, and the discovery work tends to be done by them. And then we're constantly talking with them, and we help them with that work, and then we move forward into the IND-enabling preclinical studies, and then through into clinic across our range of programs. Next slide, please. You can see here, this is our large portfolio of programs. And I apologize, it's a little hard to read, but the three colors denote three separate, what we call franchises. Now, even though they're separate, they really are interlinked. So the first franchise is our neurodegenerative disease franchise. This is a group of diseases where there is an ongoing progressive destruction and loss of neurons. And in those situations, it's really important to identify diseases early and treat early. So this includes diseases such as giant axonal neuropathy, where there's an ongoing progressive damage to neuronal architecture. It includes metabolic lysosomal storage diseases such as GM2 gangliosidosis and CLN1 Batten disease, where there is an ongoing progressive loss due to the pathophysiology of these diseases of neurons. And we have several programs in advancing forward through IND-enabling studies and into clinic in that franchise. The second franchise, which you see is in the yellow, this is what we call the neurodevelopmental diseases, and Angelman fits into this category. This is where there isn't necessarily an ongoing progressive loss of neuronal tissue, but there is some dysfunction, some disordered functioning with the brain, and we need to somehow correct that in order to ameliorate the signs and symptoms that are being exhibited by the particular patient. So Rett syndrome is another one of those conditions. Angelman, as I've mentioned, fits into that group of diseases. And then in the bottom part of the diagram in the green, you see what we call the genetic epilepsies. And historically, 20 years ago when I worked in clinical practice, we'd often identify children who start having seizures early on in life in the first few days, and there's no way of really diagnosing them. They end up being treated with steroids and antiepileptics, really without much benefit. They then go on and develop, they kind of don't develop in the right way and don't acquire milestones, and often there's a mortality before the age of 10. And more recently, we've been able to understand and describe the molecular underpinnings of many of these diseases. And there's obviously a big link with the neurodevelopmental franchise. So it's a bit of an arbitrary distinction, but at the moment, we feel it's valuable to keep those franchises a little bit separate. But this includes diseases such as SLC13A5, which is a disorder in citrate metabolism, and S6A1, which is a disorder in the gene coding for the gamma aminobutyric acid, or the GABA receptor. And we have several programs moving forward in that franchise as well. But all franchises are interlinked, and I think it's an important support to understand that we use a very similar platform for every one of our diseases, and also we can learn from each disease to inform the other disease. And it's really important to understand that, because we've had 9 regulatory meetings as a company over the past 6 months, not just with the US FDA, but with several other international agencies across our portfolio of programs. And every discussion, because the diseases and our approach is common, you learn from one to inform another. So we really believe in this platform approach to address the wide range of conditions. Next slide, please. And on that note, in terms of where we're up to specifically across the other programs, we have 3 programs in clinic currently, and we're learning from those programs to inform others. We have our giant axonal neuropathy program. We'll be sharing high-dose data from that very successful program. We've dosed 14 patients on that. We'll be sharing that towards the end of this year. I know it's close to the end of this year. We will be sharing biomarker data from our GM2 gangliosidosis program. This is also a program in clinical development. And we'll be sharing safety data from our CLN7 program, also a program in clinical development. 3 patients have been dosed in that particular program. So we are moving forward in very rapidly with clinical trials across our portfolio. We also, by the end of 2022, we'll be sharing biomarker data from our CLN1 program, likely some clinical data as well, and then clinical data from our RET program. All those programs are on track currently. Next slide, please. So that's Taysha as a whole, but now I want to touch on Angelman specifically. And we have 2 programs, 2 approaches to treating Angelman. Once again, everything is AV9, everything is HEK293, everything is being given intrathecally. But the 2 approaches are at a high level. One is where we replace the gene, and the other is where we knock down the ATS transcript, so mimicking to a degree what happens with the ASO approach. And you'll have heard already, I'm not going to go into detail with these programs, because you'll have heard already in detail from these programs from our academic partners. So Ryan Murphy from UTSW will have shared the knockdown approach, the shRNA approach. And Ben Philpott, his partner Matt Judson at UNC will have talked about the gene replacement therapy approach. Next slide, please. So with regard to the gene replacement therapy approach, and it's a little hard to see, I think, the actual construct itself, but this is a design of the construct in the lower part of the slide. So basically, we include the full-length UBE3A gene that codes for both the long and the short isoforms, and there's use of a neuronal specific promoter, and that's will be wrapped up in an AV9 capsid. And in that situation, we're replacing the UBE3A gene that has been knocked down, therefore producing the right amounts of the protein. Next slide, please. And then with Ryan's approach from UTSW, once again, we use an shRNA, so short hairpin RNA, to unsilence the anti-sense transcript and allow expression, once again, of the UBE3A gene through the paternal allele. We use a slightly different promoter in this situation. It's what's known as an RNA 3 polymerase type promoter, which is often used for these shRNA approaches. But what this should do, it should knock down the ATS transcript and allow the paternal allele to express the UBE3A. So we're using both approaches. We're advancing both approaches into clinic. And in terms of where we are with regard to timing, they're really both at the preclinical stage. We are completing proof of concept in the animal models and are moving now into talking about what IND-enabling studies will need to be done for both programs, i.e. specifically what pharmacology in the mouse model and what kind of toxicology might be needed, either in the rodent model or more likely in the NHP models. We tend to be very robust and conservative with our toxicology, i.e. we generally do 3 species of toxicology: chronic mouse tox, 6-month rat tox, and then 3 or 6-month NHP tox. Really just because there's, as you will all know, just in general terms, there's been an increasing focus on the safety of gene therapies, AAV gene therapies in recent years. Next slide, please. So I won't touch really in much detail on Angelman syndrome, as I'm sure many of you will know this disease better than I do. But the intent is to allow expression of the UBE3A gene, allow protein to develop, and therefore ameliorate the signs and symptoms. Once again, I think it is less urgent for a neurodevelopmental disease than a neurodegenerative disease to treat early and aggressively. But in general terms, for all neurological diseases, before there's been an allowance, an accumulation of disease burden, it's best to identify patients earlier and treat earlier as much as possible. Next slide, please. So in terms of the approach and where we are specifically with Angelman, discovery has occurred. So what discovery means is someone has an idea, let's try and use this idea to see if we can test the hypothesis cell lines and in early animal models to see if there might be a glimmer of a hope that it might work. And that is clearly the case both with the shRNA approach and with the gene replacement therapy approach. So we're completing those proof of principle studies and are now in the phase of designing and thinking about IND-enabling studies. When I say IND-enabling, what I mean is the preclinical studies that are needed in order to allow entering the clinic. So we're in the middle of that preclinical research phase. After that, we'll start the clinical research. We haven't publicly announced any timelines for clinical research. If I had to guess, it's probably at least a year away, dependent on how the preclinical studies progress, but we haven't been very firm about timing on this. And then, of course, once preclinical research has moved into clinical research, the studies are done, well, then the drug development approach is to think about talking to the regulators about filing the BLA. My expectation is that we're going to have some kind of scientific advice meetings with the regulators, not just in the US, but also in Europe and perhaps Canada, perhaps Australia, during the course of 2022. So 2022 is a year where we're going to be completing IND-enabling studies, having those in-depth discussions with regulators, and really preparing for and planning in detail the clinical trial plan for both programs. Our intent is to move forward with both programs to allow, at the end result, patients to have the opportunity of one or the other. Next slide, please. Well, I'll stop there. Thank you very much for allowing me the opportunity to talk a little bit about Taysha and our approach. Very happy to take questions a bit later, but in the meantime, we're going to hand over to my good friend and colleague, Emily, to tell you a bit about how we like to work with yourselves. Emily.
Suyash, and thank you for the opportunity to present today. I had hoped to be in Austin this weekend. I actually am a Longhorn. I spent many years long time ago in Austin. So unfortunately, due to a family conflicts, I couldn't be there. The other thing I miss is really getting an opportunity to meet patients and families. So again, hopefully we can meet next year at another time, and I can meet everyone in person. So that was one of the most rewarding things that I found with working with the spinal muscular atrophy community in the past, was the ability to really connect with patients and families and get a better understanding of day-to-day lives living with the disease. So look forward to learning more in the future. So I think Allison did a nice job of my background. I'm going to talk to you a little bit about how me and my team work with patient advocacy groups as well as the community. So, Taysha was founded with the idea that patients are the center of our work. And we developed these patient guiding principles. And really it starts with collaboration. As Suyash mentioned, we were founded on collaboration with UT Southwestern, but then we also closely collaborate with advocacy partners. And Allison from FAST has been a phenomenal partner early on, sharing the non-competitive information that really is helping us in preclinical studies. But we will continue to collaborate as we move forward with these programs. And I'll touch on some of those areas in a bit. We also believe in transparent communication. So as much as possible, we want to keep the communities up to speed on the latest information and where we're at with our gene therapy programs. And we also approach things with curiosity. We can read all the literature that's out there, but there's always a lot of questions that come up. And honestly, we believe that our advocacy partners and patients and caregivers are the experts in this area. So we do spend time learning directly from our advocacy partners and patients and families. We also approach things with empathy. Just the way that we work, we interview people for that as well, regardless of the program. It's one of our core values as an organization. And lastly, respect. Oftentimes we have the opportunity to learn a lot about patients and families, and we have strict confidentiality rules that we abide by as well. So I mentioned this a bit. Not only do we have a partnership with UT Southwestern, but we also believe that our advocacy partners play such an integral role in developing the treatments. And already we've gained such fantastic insights from FAST, and I know that this collaboration will continue. As I mentioned, there are a number of ways that we'll partner with FAST to bring a gene therapy to families. And first is really understanding the unmet needs of patients and families. And this helps us as we're developing our clinical trial programs. What are the endpoints that matter the most for families? When developing a clinical trial, how do we need to think about setting up the evaluations for patients and families? What are the needs from travel? What are the needs at a local level as well? We also want to identify educational gaps for not only families, but clinicians. We want to, oftentimes in my past, we learned that there was a need to educate more on a gene therapy program. So we'll really try to get a sense of where do we need to educate patients and families in order to feel comfortable participating in a clinical trial and allow them to really weigh the benefits of a potential program. We also lean on FAST and other advocacy groups to be our disease experts. I think I mentioned that earlier. You all are the experts. We can read the literature, but we want to learn directly from you. I mentioned, obviously that the insights that we gain will lead to how we develop our outcome measures for our clinical trials. And then this should also help us think about how we can enroll patients as well. And then I've also found, we found that again, our advocacy partners are the experts, and they can tell us what clinicians should we be talking to, where are the researchers at, where should we think about setting up sites, and that's a great partnership. And then lastly, we work to co-create educational resources with patients and families as well. So again, if we understand that there's a gap in education, we'll then make sure we're working directly with the community to make sure that those educational programs and materials meet the patients and families' needs. I think this just reiterates our partnership here. We really want to thank not only our collaborators at UT Southwestern, the UNC School of Medicine, our colleagues at Taysha, the Foundation for Angelman Syndrome Therapeutics, as well as the Angelman Syndrome Foundation and the entire Angelman community. And we really look forward to partnering with you to advance our programs for Angelman. Thanks so much.